Welcome back, guys. So in the first part of this video, that is the life cycle of Puccinia, I had explained the uridospore, the telutospore. Okay, so from here we will continue with the basidiospore. Now the basidiospore, I had mentioned how it forms, how the telutospore it germinates. So it developed on the germination of the telutospore. Okay, you can see here the telutospore when it germinates. So through the germ pore, you can see here the two germ pore. The germ tube will emerge through these germ pore, and what will happen? The spore will move. The nucleus, the diploid nucleus will move to this germ pore and it will divide through meiosis to form four haploid nuclei. These four haploid nuclei, they are of two different types, and two of them they are of positive type, and two of these nuclei they are of negative type, and also a wall formation will start to form. So here later on, in this case, in this figure, see this germ tube now is referred to as probasidium or epibasidium. Okay, and it has a limited growth. So what happened later on? The probasidium will become four cell. You can see here it has become four cell through the wall formation. Okay, four cell by transverse wall. Now each wall, you can see here, it develops a small projection. Okay, it will develop a small projection, that is the sterigma. It is known as a sterigma, through which both the nucleus as well as the cytoplasm will push to this projection, will push outside, and it will develop basidiospores. So you can see here the sterigma, the small projection will form, and the nuclei and cytoplasm will push outside and forming a basidiospore. Now we have four basidiospores, two types of which are positive and two types of negative. So from all these telutospore, the basidiospore will form, okay, through the germination of all these telutospore. Now many basidiospores will form. Now the basidiospores, if you just take a look at the basidiospores, they are unicellular and also thin walled and very, very small. So after they get discharged from the basidium, okay, the basidium, after they get discharged, these spore, they are disseminated by the air current. Okay, so how they get discharged? It is by explosive mechanism. So by explosive mechanism, the basidiospores, they are discharged from the basidium and they are disseminated by air current, by the wind. So what happened to these basidiospores? These basidiospores, they can survive only for few days. Okay, they can infect only the leaves of the alternate host, that is the barberry, not the wheat plant. Okay, so now the basidiospores, after they are carried away by the wind, if they come in contact with the alternate host, then only they will germinate. But in case they do not come, those spores which cannot come in contact with the barberry plant, that is the alternate host, they will eventually die. Okay, they will die. Now, so those basidiospores, after they come in contact with the barberry plant, they will start to germinate, forming another type of spore, that is the pycnospores. Here you can see these basidiospores which already get detached from the basidium. Okay, they are round and they have a thin wall, they are unicellular and small, and also they are haploid. Remember, because the spore here, it has already, the nuclei, it has already undergone meiosis to form four haploid nuclei. So these basidiospores, they are haploid. Now when they come in contact with the host plant, that is the barberry, the alternate host, they will start to form another type of spores, the pycnospores. Now, during favorable condition, the basidiospore, they germinate on contact with a barberry leaflet towards the upper surface. Suppose this is a barberry leaflet. You can see here these are the pycnospores. Okay, the pycnospores, they are also known as spermatophore. They they form, okay, they germinate towards the upper side, okay, the upper side of the leaflet, now producing the germ tube. Okay, they will produce germ tube. Now the germ tube, it will penetrate the epidermis, and grow there intercellularly. Now the nature of the mycelium, it depends on the nature of these basidiospores. Okay, what does it mean by that? Because we know that the basidiospores, they are of positive as well as negative types. So the nature of these pycnospores, it depends on these spores which come in contact with the host plant. So here, suppose these positive type will come in contact with the host plant. Here in this part, the pycnospores will form and they are of positive type. And here, because the negative type basidiospores, they come and germinate here, so these are of negative type. So within a few days, the growing mycelium, it will become aggregated under the epidermis. Okay, here under the epidermis, it will become aggregated and forms a yellowish flask shaped structure. You can see here, this is like a flask. Okay, a flask shaped structure, and this flask shaped structure, it is known as the pycnium. Okay, the pycnium or the spermatogonium. The pycnium or the spermatogonium, this flask-shaped structure. Now the pycnium, it has a small sterile mycelium on the neck. On this neck part, it has a small sterile mycelium called the periphysis. Okay, called the periphysis. Now these periphyses, they intermingle with much larger thin-walled, simple and branched receptive or flexuous hyphae. You can see here these two, this one, these are the flexuous or the receptive hypha. Okay, you can see these flexuous and receptive hypha which are formed and which intermingle with the mycelium. Okay, so just at the neck of this flask-shaped structure, that is the pycnium, you can see the presence of these receptive hypha. Now, what is the function of these? I will tell you later. So, the bottom of the inner side of the pycnium is lined by many uninucleate tapering cells. You can see here these tapering cells here. So these many uninucleate tapering cells are lined in the inner side of the pycnium. Now tapering cells, these tapering cells which are lined here below, they are referred to as the spermatophores, okay, or the pycnophores. The pycnospores or the spermatophores which develop. Now it is from these cell through which the small oval spherical uninucleate cells will develop, and these small spherical uninucleate cells, they are referred to as the spermatia or the pycnospores. So these are the pycnospores, uninucleate cells. Now depending on the nature of the basidiospore, so the pycnium and the pycnospores also, they may be of positive or negative type. Like for example, as I said, that suppose if the positive type, it falls here, this pycnium will be of positive type, and the pycnophores which are formed, they are of positive type, whereas these will be of negative type. Now, numerous pycnia of different types, they grow in cluster. Okay, they grow in cluster. You can see here many. They grow in cluster on the surface of the leaf. Now, the pycnospore or the spermatium, it does not infect any host. They remained here on the barberry plant. They did not, these pycnospore, they will not infect any host. So the major pycnium, both this positive and the negative type, what happened, they actually secrete a nectar drop. Remember this. Okay, they will secrete a nectar drop during release of the major pycnospores. You can see here the pycnospores, they are released outside, and actually this pycnia, it will release a nectar drop during this time. And this nectar drop, what will happen, suppose an insect comes in contact with this leaf, barberry leaf, so the insect, it gets attracted by the nectar and help in the transfer of the pycnospore to the fleshless hyphae. Now what will happen, suppose an insect, it comes in contact with this pycnia here, so the positive spore will get attached to the feet of the insect. Now if that insect, it comes in contact with this negative type, so those spore, they will come in contact with this receptive hypha. So the positive spore will come in contact with this negative fleshless hypha or receptive hypha. Okay, so let me show you another picture. Yes, you can see here very clearly this figure. You can see this is the ruptured host epidermis. The spores are releasing outside, and these are the single nucleate cells which are aligned in a ring known as the spermatophore or the pycnophores, and you can see these are the receptive hypha. What is the function of this receptive hypha? You can see here what happened. The receptive hypha, you can see here, suppose now from the negative type, the spore comes in contact with the positive type, the receptive hypha. So these positive receptive hypha with this negative spore, pycnospores, they will fuse together. Okay, they will fuse together in a process known as spermatization. You can see this receptive hypha, it's coming from here. Suppose these, everything, it happens here. Now this is the receptive hypha from here, whereas the spore, it comes from this one. So how? Due to insects. Okay, when the insects, they are attracted by the nectar drops on these barberry leaf, okay, they will help in the dispersal, okay, in the transfer of the pycnophores from one receptive hypha to another. Okay, so what happened when they come in contact like this? They will fuse together. So the transfer of pycnophores to the fleshless hypha of the opposite mating type, that is the positive with the negative fleshless hypha. Now the wall, okay, what happened here when they come in contact together? The wall here, it will start to dissolve. Okay, it will dissolve. The wall at this point of contact, it will dissolve, and the nucleus of this pycnophores, okay, here in this case it's positive, this nucleus, it will pass through this fleshless hyphae. Okay, it will pass through this fleshless hyphae. Now what happened during that time? A dikaryotic condition is established. Okay, when a negative and a positive, it will fuse together, the nucleus. So here we have a dikaryotic condition. Okay, so a dikaryotic condition is established, and this process is known as spermatization. Now another type of spore actually will develop on the same plant, that is the barberry plant. Okay, so wherever these pycnophores develop, the archaeospores also will develop. How? Because these archaeospores, they are formed from these pycnospores, okay, during development. Now what happened here after spermatization? Okay, when we have the dikaryotic mycelium, now during development of the pycnia, some hyphae of positive and negative type, they goes downward. You can see here these, okay, the hyphal connections. Now the hyphae will go downwards. Okay, they will go downward towards the lower side of the leaf. They go downwards towards the lower side of the leaf, forming a respective immature Asia. Okay, this is Asia, or we call it acial primordium. So further growth is initiated within few days of spermatization. Okay, further growth is initiated within few days of spermatization. Now the spermatial nuclei, after they fuse together, so that nuclei, that's spermatial nuclei, will passes downward through these hyphae. Okay, it will passes downward, and after crossing the inner tissue, they reach and dikaryotize the basal cell of the acial primordium. Okay, they will dikaryotize the basal cell of this acial primordium. Now this acial primordium here then stimulate to further growth within few days on the lower surface of the leaf. You can see here the acium, okay, it is this clustered cup, okay, is formed. You can see here it's like a cup, an inverted cup, known as the acial cup. Okay, this one, it's like a cup. So here this will form, and this young acium is actually buried inside. Okay, it is buried inside the tissue just below the lower epidermis. But what happened with maturity? Okay, when it becomes matured, it pushes and nurtures the epidermis, thereby the spores become exposed. Okay, so first it is buried in the lower epidermis itself, but after maturity, when due to the spore formation, this will ruptures the epidermis and the spores are exposed to the outside. Now the acium here, just remember, this is an inverted cup shaped structure. So the pycnia are formed on the upper surface, whereas the acium will form on the lower epidermis. Now you can see here the outer margin. You can see here in this cup-shaped structure, this outer margin is known as the peridium. So the stalk cell, after become dikaryotize, they divided mitotically into a chain of alternately arranged large and small cells. Just take a look at these, the enlarged picture of these you can see. Here they divided mitotically into a chain of alternately large and small cell. Now the large cell, these large cell here, they will form the archaeospore. Okay, they are dikaryotic. You can see here two nuclei. So they are dikaryotic cell spores, and these large cell, they will form the archaeospore, whereas these smaller one, these very smaller one here, they will form the, they will become sterile. Okay, they will become sterile, and they are known as the disjunctor cells. So the disjunctor cell, what is the function of these disjunctor cells? Actually, these disjunctor cells, they help in the dispersal of the archaeospores. So the big one will become the archaeospores, the smaller cell are the disjunctor cell. They will become sterile, but they will help the archaeospore. They will help in the dispersion of the archaeospore. You can see both all the cells here, they are dikaryotic. So the archaeospores, they are actually unicellular and binucleate, okay, with two nuclei, thin wall, and also they are orange in color. Okay, they are orange in color. And the young archaeospores, just remember the structure, they are polyhedral in shape. Okay, they are actually polyhedral in shape, but they will become globose. They will become globose and round with maturity. So these archaeospores, when they are released to the outside environment, they are dispersed by the air current, and they can infect only the graminaceous host, that is the wheat plant. Okay, they can infect only the primary host, okay, the wheat plant. So after falling on the wheat plant, the stem or the leaf, they germinate by producing germ tube, and that will penetrate inside the host and establish infection. I will just show you again this picture here, this one. See, archaeospores, they have already come in contact with the host, that is the wheat plant, either the stem or the leaf. So they will start forming the germ tube, and they will grow intercellularly, forming the secondary germ tube or the dikaryotic hyphae. Okay, so that those are the different types, 5 different types of spores produced in the life cycle of fungi, of this Puccinia. Now you can see here, this is a microscopic slide showing these inverted cup shape. They are the acial cup shape. They look like an inverted cup, and these are the spores. You can see this is the infected barberry plant, showing the leaf with the pycnia. Okay, the upper part you can see here, the yellowish pycnium. Okay, they are the pycnia on which the pycniospores are formed. So if you take a look at the overall life cycle of Puccinia, what do we see here? Suppose this is the not a grass, okay, suppose this is the wheat or a grass host. Okay, so what is its life cycle is that it will start to form uridosauri. So from these uridosauri we will get the uridospores, and these uridospores during formation, during winter season, okay, in favorable condition, this process will repeat again and again. They will germinate, but at the end of the wheat season, they will start to form telutospores. Okay, telutospores, they will germinate to form the basidium, the basidiospores, and this basidiospore will infect the alternate host, that is the berberis. Okay, the berberis vulgaris, that is barberry plant, and here they will germinate to form the pycniospores on the upper part and the archaeospores on the lower surface, and these archaeospores, they will in turn infect another wheat plant. Okay, through wind dissemination they will come in contact with the wheat plant and they will infect the wheat plant. So this is the graphic life cycle of Puccinia graminis tritici. You can see here how the you can see here the acidiospores. Okay, when they come in contact and germinate on wheat plant, they will form the uridocerus, which will produce uridospore, and this process, it can proceed, it can multiply, it can happen several times here. This is the dikaryophase. Okay, why? Because we have the dikaryotic mycelium will form, and then from the uridospore the telutospore will form, which will fuse, the nuclei will fuse together. Okay, karyogamy, and this telutospore on germination and through meiosis it will form 4 haploid nuclei. Okay, 4 haploid spores, that is the basidospores, 2 of which are positive and 2 of negative type. Then these on germinating, when they come in contact with the barberry plant, okay, they will form 2 types of mycelium. Why 2 types? Meaning positive and negative type. They will either form a positive and negative type depending on the basidospores which come in contact with the barberry plant. So in case of positive type, we will have a positive pycnospores, okay, which will come in contact with the negative receptive hyphae, okay, through the help of insect. And also in monokaryotic mycelium negative type, okay, we will have a negative receptive hyphae. This negative receptive hyphae, it will come in contact with this positive pycnospores. So in both the case, if spermatization happens, it will develop a dikaryotic mycelium, and these will form the acidyl cups, which will form the archaeospores. So these things, they happen on the barberry plant. Okay, the formation of the pycnospores and archaeospores, they happen on barberry plant. Whereas the telutospore formation and the uridospore, they form on the wheat plant.